I worked in this area of nanosuspensions and micro channels for a number of years, and we find new applications every day. I worked in Knowles Atomic Power Lab for 10 years. Much of that work is ITAR, and I can't talk about it, but there again, I was working in the cooling channels of reactors, which are very specific in shape. But today, I want to talk about microchannels and droplets, and specifically the electrified micro droplets. And here the fundamental aspects, my group focuses on three things, and the fundamental aspects of microchannels and droplets that are about 20 to 100 microns in size. These channels can be used for cooling and transport properties. They can carry nanoparticles to areas that are not easy to do otherwise. You can use it for sintering, and so there are new, and later on I'll be also talking about how we can trap viruses potentially with this. Moving on to, I want to also mention that I have on the right side, let me see if I can get my, yeah. Here on the right side, and you can see that I've got long-term collaborations with Dr. Carr, and I've also working with Dr. Davis in this particular patented technology called nano electrospray laser deposition, or also called NELD. And later on, I'll be talking about semiconductor nanoparticle structures for trapping. I'm initiating that new project with Dr. Kaur and Dr. Manish Gupta of College of Medicine, and also have worked in the past with Dr. Cho and MAE on microfluidic devices, and currently working with Dr. Bhattacharya and MAE in devising new channels. So the work consists of levitation. Droplets can be manipulated to do all sorts of things. One of the things that you can do is to electrify the micro droplet microfluidic networks. Here you can see that by judiciously keeping the electrodes in channels, you can create the electric field that is varying, as you can see from the CFD simulation with Maxwell equations. By increasing the voltage in those electrodes, you can either slow down the droplet, channel to the position where you want it to go, and downright pin it to the wall if you so desire for whatever application you have. You can also, in the same, this is done by one of my students in the last 4 or 5 years, and his name is John Waking, published this in Lab on a Chip. Here in this particular case, you have two different types of droplets coming in, and it will allow the small droplet in a certain channel, and the big droplet will not be allowed in, but the electric field would take it to the channel that you want it to go to. This allows these throughput to be increased, and a frequency of these droplets to increase, so that those droplets can then be, you can create powders of the same size, uniform size, which is very difficult to do. So using these techniques, we have done the experiment with indium electrode, and found that we can actually completely deform the droplet and pin it, or exactly as shown in the simulation. And when you turn off the electricity, then you get the regular droplet back. So this allows us to do a few things, particularly in a second phase can be introduced with a chemical compound. You can see that for drug delivery, this could very well be used in terms of creating capsule. And at this stage, it is in droplet. But then beyond this position, you can actually make it solidify also, which is what I want to show next. So you can solidify the droplet using heat here, and this is a time-delayed chemical reaction in a droplet, which has this alumina in this particular case, but there are other compounds that you could use as well. These microchannels of typically 100 microns, they carry droplets, they carry droplets of the same size as smaller size. But more importantly, these droplets are carriers of nanoparticles that are 10 to 30 nanometers in size. And so, see, it's not easy to transport nanoparticles, but by carrying it through droplets, you can transport them. And you can increase the concentration of the particles in a suspension. And so we've used these combined effects of nanosuspension of high viscosity alumina. The viscosity keeps increasing, as you can see in this blue line, that first, as in liquid phase, it actually comes down with temperature, but beyond a certain temperature, at the point of solidification, it starts increasing the viscosity. And that, so the heating and the agglomeration characteristics of alumina inside the droplet will produce uniform spheres, as you can see on the right-hand side, as we have demonstrated experimentally in a microchannel. And these powder form are actually 30 microns in size. This was published in APL by the same student and also another student named James Wilson, who did his honors thesis in this project. Moving on to using these electrified channels in other areas, I want to show you two pictures that you might have heard about a couple of weeks ago from Dr. Carr in Creo. And he and I have collaborated on this for almost 5 years now. And the bottom line is that in a simple way, you can explain this as water micro droplets with nanoparticles are transported to the substrate in an annular space of the hollow beam right here. This is the annular space. And the laser beam then evaporates the liquid and deposits the particles to form a nanostructure on the substrate. The substrate can be flexible. It can be a solid silicon substrate, or it can be a cardboard. And we have done all of them. And you can see how the lasers come in there and evaporates the droplet one at a time. The droplet's size can be increased. So all these, there are multiple parameters, and I'm just simplifying it to kind of give the principle. So using this principle, two of my students, Eduardo Castillo, who has graduated, and Tiani Lee, who is going to be graduating very soon, we are using this technique to actually come up with some idea on how possibly to or potentially to trap viruses. So the droplet acts as both carrier of nanoparticles as well as superlens, and it can potentially trap human viruses like SARS-CoV-2 and other viruses as well, rhinovirus, etc. See, these viruses have different protein and lipid layers carrying different charges and polarity. That's the concept here. So we'll tailor the P and N type nanoparticle clusters that will attract, trap, and encapsulate the virus. So the hypothesis is that positively charged particles will be attracted to the negatively charged glycoproteins, that those spikes that you see on in many figures that you have seen with COVID on the virion, and encapsulate the particle, so it blocks its ability to attach to an infected cell. So this is our hypothesis. We are now internally funded, and we're going to kind of get some preliminary data on this, and hopefully write an NSF and NIH proposal as well. So the nanoparticles we are considering, so far we have considered only one type of nanoparticle to do this, but now you can have two injectors, as shown in this figure right around here, two of them, and get them in a canister, and then try to get them through this hole in the mirror, and in midair you can actually sinter it and collect it in the biofluid down there. Okay, so the particle itself, we haven't actually developed what type of particle we'd use, but we got some idea. It could be an insulator like silica, or a semiconductor conductor like silicon or germanium, or a gold and tantalum like metallic particles. So we'll explore which type of cluster will encapsulate the specific virus, and we have an innovative idea to use these syringe pumps to fuse the particles and collect it in phospholipid, which is a biofluid down there. Once we do this, then we'll send it to the virology lab. The potency of the nanocluster will be tested then by Dr. Gupta, and for infectivity on sensitive cell lines and controlled tissue culture system. So we've got this IR1 on this internal funding, seed funding, and we are working on this to put this together in the form of a proposal in the next 2 or 3 months. Don't know how much time I have, but I want to talk about this new project again that we are initiating with Dr. Bhattacharya in simulating blockage in arteries. And this is typically a Mohs artery on the left that you are seeing, a normal artery on the left, and on the right you see what is called atherosclerosis. The atherosclerosis is a lot of people in College of Medicine are studying this. I don't know a whole lot about it, this biology part of it, but I know that it's a disease of the arteries characterized by the deposition of plaques of fatty material on the inner wall. So, from our end, what we are doing currently is to kind of simulate this by getting a velocity field near the branching region of the artery. You can see the circle right around here, and that is the simulated Mohs artery over there. And you can actually take pictures of that, of the flow. What we have done is using one of the droplet generation techniques. We are currently using silicon oil. Very large droplets is not like cells that are agglomerating and creating plaque, but we're trying to demonstrate this in a slightly larger channel of 400 microns, 500 microns, and so on, and show blockage, but more importantly, show on the right-hand side right here, this is micro PIV. We can get as low as 100 microns a second, even a lot smaller than that, to show how the particles move, how the fluid flow goes, and where it accelerates, so that we can use this for a proper to improve the drug delivery techniques and so on. And finally, on the right-hand side, you see that this is the long stem of the artery here, which is very large. Okay, 700 microns. We are trying to make it smaller, but the innovative part of it is our student Brian Palogan is actually come up with an actuator that actually changes that size from 700 microns to 500 microns, and so that you can create a sinusoidal flow and replicate the blood flow in there. Finally, I want to show that there is this encapsulated cell therapy is very important to cure cancer. And my student who just graduated, Mohammed, he worked in this area in terms of characterizing different modes of this encapsulation technique. And what he had found recently and published in scientific reports is that if you have a viscoelastic shell material around the cell, as it goes through the microcapillaries and so on, the higher the viscoelastic substance, the more the cell viability is. And this is an important work that will be continued, and our group is slowly moving in the area of cell dynamics and so on, both computationally and experimentally. So I want to kind of stop here and acknowledge my many students over the years. There have been so many who have worked in my lab, my collaborators, postdocs, and several students who have written multiple papers and that have been cited quite well. And the path for my students to become faculty is to first become a postdoc. So a couple of them, Abhishek and Amit, have gone to Michigan and Princeton, and from there on they moved on to better schools. And I had more than 25 students, and only 4 of them. I tell all of them that this is the greatest job in the world, to become a professor. some of them actually believe it, and so 4 of them have become professors, and now 4 more will be postdocs, and hopefully many of them will be professors as well. So thank you very much, and I'll stop here at this point.